Pui-Shee. She is a PhD student in Dr. Leanne Godino's lab, and she at the Technical University of Munich. And she's going to be talking about the formation and maintenance of horizontal cell mosaics in the zebrafish retina. The stage is yours, Pui-Shee.
Can you give me 1 second? Can you see my screen or it's not visible? Okay. So basically, as Leena mentioned, I'm going to talk to you about the formation and maintenance of horizontal cell mosaics in the zebrafish retina. So first of all, maybe most of us here know more or less about the general architecture of zebrafish retina, or in general vertebrate retina. We have the retinal ganglion cells that projects to the brain for visual processing. We have the photoreceptors that detect the light, and we also have interneurons such as horizontal cells, bipolar cells, amacrine cells. And we also have glial cells called Muller glia. So basically during development, the retina is essentially an apical basal oriented element, which then gradually transform into a structure with characteristic cellular pattern. So basically at about 30 hours post-fertilization, there are still apical basally oriented elements. And then at about 3 days, you can see the layer structure with all the different layers of cells. And however, if you actually slice through one of these layers, what you can find is the somata of these cells. And many previous studies have shown that these arrangements are non-random. So this is an example of how the zebrafish cone mosaics looks like. The 4 different colors indicate the 4 different cone types that zebrafish has. And you can clearly see that they are sort of non-random. And a lot of studies has been looking at how the layers are formed, but very little is known about how the mosaics are formed during development. So this is what I'm focusing on. So what is a retinal mosaics? It is non-random arrangements of cell bodies of individual neuronal subtypes. And it is first quantitatively analyzed by Wassel and Riemann in 1978. Functionally, it is proposed that it is important to ensure that all parts of the visual field have access to a full set of processing elements, so that you can get parallel processing of visual stimuli from all receptive field. And there's also a theoretical work that showed essentially that photoreceptors mosaics is designed to optimize visual performance. Regular arrangements can also help in developmental wiring of connection between cell types. So mosaics are independent of each other. So each mosaics of different type of neurons are independent to each other. And mosaics and dendritic toweling can be independent of each other. So one is about the arrangement of the somata, and the other is the arrangement of the dendrites. So basically, I'm looking at horizontal cells. So a brief introduction of what horizontal cells is inhibitory GABAergic neurons, and it is shown to be important in contrast enhancement and the generation of antagonistic centers around receptive field. And in zebrafish, horizontal cells can be classified into 4 distinct morphological subtypes with unique connectivity to photoreceptors. So I'm going to focus on just one subtype, the H1 subtype. This is how a typical H1 cells look like on fast view. So this is how the photoreceptor look at the horizontal cells. It has an axons, and the dendritic tips actually terminates in a roseate flower shape. And it can be labeled genetically in a specific manner. And it is shown to be connected to red, green, and blue cone. So first of all, to ask the question of whether there is a mosaic that is a H1 mosaic, I basically dissect the retina out of the animals, and I flatten it to make a flat mouse. So we know that by 3 days there's layers. So I'm asking whether at 3 days I can see a regular pattern. This is a representative image of how the retina look like, and you can more or less see that there is some sort of arrangement here. And I basically do automated segmentation and then analyze using spatial statistic analysis. There are 3 methods that I use. The first one is Nearest Neighbor Regularity Index. It's only concerning about the nearest neighbor. Neuronal domain regularity index, basically it is assessing how the whole retina space is spaced out by different cells. And that's also a density recovery profile. It is more about looking at the global arrangement. So using all 3 methods, comparing to random simulations, I basically see that H1 mosaics is established as early as 3 days, because for all of the index that I show, it is always higher than the simulation case. And how does then this regular arrangement arise during development? This is this is when I turn into in vivo imaging of mosaics formation. So it has never been done before because of technical issue, and so most of the studies is done more theoretically. And yes, so as I mentioned before, horizontal cells are generated starting at 2 days and forming a layer by 3 days. So I did a time-lapse movie from 46 hours post-fertilization to 72 hours post-fertilization, and this is a representative image. So what you can see here is that there are a lot of cells coming, and basically it follows a developmental wave of how the retina is formed. So basically from preliminary results, I can say that mosaics formation could be divided into at least 3 phases. That's 1 active proliferation, migration, and refinement stage. There are a lot of possible mechanisms as to what could be underlying neuronal mosaics formation, and I'm only going to focus on tangential cell-cell interaction, mediating tangential migration and peripheral translocation. So in the migration phase, if you look at this movie, you can sort of see that maybe I can play this again. So you can see that there are 2 cells that touch each other, and then the cells kind of move away from space into a more empty space. So basically here you can see that cells move away from crowded space, and it kind of sample the space to find where the maximum distance to neighboring cells is. And also in the refinement stage, this is a 2 hours movie of one part of the retina, and basically I do automated segmentation and Bayesian tracking. So what you can see here is the velocity of all the cells at all time point, and you can see that they have quite low velocity at this stage. And when I plotted the distance from the first time point, you can see that cells are still sort of deviated from their initial time point, but it always have this jiggle. So this poses that these cells are basically sampling the space and then find kind of where it wants to be at the end. And this can also be looked at by the membrane labeling. And you can see all the protrusion that just keep coming up and go. So all this information implies that there might be some cell-cell interaction mediating mosaics formation. So As a summary, I told you that H1 mosaic is established as early as 3 days post-fertilization, and I have established an imaging paradigm for in vivo visualization of mosaics formation, and it follows a developmental wave. And it can be divided into at least 3 phases: proliferation, migration, and refinement phase. And H1 cells are sort of actively sensing the space during migration and refinement phase. And yeah, the future direction will be to perform like sort of ablation studies to further test this hypothesis. And for that, I would like to thank you all for your attention. I'm happy to take any questions.
Thank you, Pui-Shee. That was really cool. Especially the fact that you're able to image across development and look at those cell-cell interaction dynamics across all these developmental stages. So while we wait for some questions, I have a question about at what stage do you see these cell-cell interactions? I mean, is it only after the proliferation phase when all the cells have already been born and proliferated, and then you start seeing these cell-cell interactions that push the cells away to their final destinations? Or do they start interacting with each other even while they are migrating and proliferating to find their optimal positions, even before new cells come in, essentially?
Yes, so basically, I didn't show it here, but I have preliminary results that basically show if I look at the division angles of neighboring progenitor, that don't sort of space out from each other. So it means that the progenitor doesn't know that needs to stay away from the other cells. And also, if you look at this movie, you can see that there are a few cells that sort of clump together. So it means that, yeah, basically at this phase, there's no more division at this part of the retina. So yes, the different phase.
Sounds good. We have a question from Yunlu Ju, and he says, do you think contact inhibition, CIL, could be a potential mechanism for their migration?
Yes. So there have been theoretical works that show contact inhibition could lead to a regular pattern. And yeah, one of the problems here is that we doesn't know what kind of cell surface molecules are expressed in this type of cells. So we have no way, and at least at the moment, no way to perform a sort of loss of function study to look at this. But it can definitely be a mechanism.
Related to that question, actually, I was wondering if these cells have, in addition to just contacting each other, do they signal to each other as well in terms of activity and that guides where they would migrate to, or is it purely a structural based interaction?
I haven't looked at the activity of these cells. And yes, it could be a very interesting direction to go. Definitely.
Okay, sounds good. Let's see if we have some more questions. If the audience has questions, please feel free to post them in the chat for Pui-Shee. I actually was wondering also about, you said you haven't done it, but do you have any hypotheses on what kind of cell-cell interactions might take place in terms of whether they're cell-cell adhesion molecules that interact with each other, or is it some other type of signaling mechanism?
So basically, cell-cell adhesion molecule, for example, cadherin could be possible. And also because horizontal cells does form gap junction with each other at a later stage. So that might also be one of the mechanism where you have connexins that basically bind to each other or repel each other.
And this is different, and these are only cell body tilings, right? This is different from the dendritic, like you mentioned,
Yes.
mechanisms that would probably have other kinds of dscan like molecules or something like that that probably govern those mechanisms.
Yeah.
All right, cool.
Okay.
Well,
Thank
thank
you.
you so much, Pui-Shee, for that very insightful talk. We are going to move to our final